Hello YouTube, this is Sam from Tigers Schooling, and in this session or the next lecture we're going to talk about the classification of enzymes. So classification, we have already talked about the enzyme nomenclature in the previous video, but now we're going to talk specifically about the classification. So in the classification we have got, in the classification of enzyme we have actually got 6 main classes of the enzyme, which could be remembered by the formula OTLEL. You are not allowed to shuffle this formula because this is made on the sequence. So this sequence, if you remember, you are easy to remember the serial number. You don't want to mess up the serial number or the EC number. Sorry, it was not serial number, the EC number. You don't want to mess it, because if it is 2, it's always transferase. If you move that to that one, it's going to put everything out of the system. So you have got to follow this rule. So in the class of the classification of enzyme, the first class is Oxido Reductase. The second one transferases. The third one hydrolysis. Fourth one is lysases. Fifth is isomerases. And in the sixth one it is ligases. So the word coming to the first class, it is Oxido Reductases. So this first class, the word oxido means oxidation. The reducto means reductation. The word is added because it's an enzyme. So how we can define it? So those enzymes which are involved in the oxidation and reduction of their substrate are called oxido reductase. Get it? Again, oxido reductase enzymes involved in oxidation and reduction of their substrate. Alright, we have got few examples regarding the oxido reductase. We have got the alcohol dehydrogenase. We have got one example, that is the alcohol sorry about my writing, genases. Alright, I'm sorry, just get it, it's alcohol hydrogenases. And we have got other example as the lactate dehydrogenases, xanthine oxidases. And one thing more, we have an example of the oxido reductases. We have got phenylalanine. Oxygen is adding up. We are using the hydroxylase, an enzyme which is an oxido reductase enzyme, and which converts the phenylalanine to the tyrosine. The best example I could find. And the second class we have got the transferases. So transfer, the word transfer, you get it, right? Something is being transferred. So in the case of the transferase, what we mean, the enzyme that catalyzed transfer of a particular group from one substrate to another substrate. This example is going to explain what I mean. Get the definition first. Enzyme that catalyzed transfer of a particular group from one substrate to another. Now let's move to the example, then I'm going to explain this one. We have got few examples, that is, alright. So example of that is aspartate. So aspartate, and we have got other examples, there's alanine transaminase, the best example, which is called AST or ALT. Best example I could find, ALT or AST, which is called the alanine transaminase. So this example explains it better in an easy way. So we have got glucose here, right? So glucose has to be converted to glucose 6-phosphate. So from where we getting the phosphate group? We are getting the phosphate group from another substrate. So this is donating. So the phosphate group is actually transferring from the ATP, adenosine triphosphate, into the glucose, which actually converts into the glucose 6-phosphate by the help of an enzyme which is a transferase called the hexokinases or the glucokinases. I hope I cleared the idea regarding the transferases. Now let's move to talk about the hydrolysis. So hydrolysis, you have probably heard of break down something with the addition of the water. So this is easy concept to understand. So enzymes that bring about hydrolysis are some of the examples. We have got glucose 6-phosphatase, pepsin, trypsin, esterases, and glycoside hydrolysis. Now move toward the number 4 class of the enzyme. We have got lysases. Lysases is similar to the hydrolysis, but the only difference is we're not going to add water, but we are going to break it down. So no addition of water, but breaking it down. So lysases is the enzyme that facilitated removal of small molecule or breaking that molecule from a large substrate. We have got example in this case of fumarases and the histidine decarboxylases. These are the example of the ligases. But in the case of isomerases, which is the fifth class of the enzyme, which is very important, because in the case of the isomerases, enzyme is involved in isomerization of the substrate. We have a substrate called glucose 6-phosphate. Another substrate is that is going to be converted into the fructose 6-phosphate. So for that, what we need an enzyme which actually convert one isomer of a substrate to another. So in the case of this case, we have got phosphanase isomerases. Alright, coming to the final class, we have got the ligases. Ligases means joining something together. So ligases are the enzyme that are involved in joining together two substrate. Example, we have some example regarding this one is the RNA synthase and the DNA ligases. So this is a pretty easy concept to understand. I hope you get the whole idea regarding the classification of enzyme. I'm going to make a detailed video about the classification of enzyme in coming soon, because it's not the detail, it's just a brief description of the classification enzyme. I hope you have a good day. Keep visiting Tiger Schooling. Thank you.